My name is Mariana España, and with my friend Maria Jose Varela, we are going to present our genetic modified that is polydactyl.
Genetic diagnostic card. Case description. Polydactyly is a condition where a baby is born with extra fingers or toes. It can happen in one hand or foot, or in body. Some babies only have this, but others can have other health problems too. In some study from Chile, doctors found 92 babies with polydactyly out, out of more than 37,000. Most of them had one extra finger or toe, one outside part of the hand or foot. It was a title more common in boys, and sometimes other people in family also had it, which means it can be inherited. Polydactyly was found to be caused by mutation on the short arm of chromosome 7 in the GL13 gene on the another identified on the PAPA. Additionally, another was identified on the long arm of chromosome 13 and in the PAPA3 gene of the short arm of chromosome 19.
Problem question. Why do some babies have extra fingers or toes when they are born? And does it come from their parents? Hypothesis. If a baby has chains and its genes or has parents with extra fingers or toes, then the baby can also be born with extra fingers or toes. Variables. Independent variable. Genetic change or if parents have extra fingers or toes. Dependent variable. The baby being born with extra fingers or toes. Controlled variable. Type of study. Number of babies observed and where they were born.
Strategies used to collect data. Data-based use. Hospital Clínico de la Universidad de Chile, 37,008 births analyzed in 1991, 2,092 cases of polydactyly identified, 64 isolated, and 28 associated.
Strategies to collect data. Review of clinical birth and genetic records. Collection of family history to allay hereditary patterns. Classification by type of polydactyly, pretextual or postdactyl. Cases grouped as isolated or associated with other malformations.
Data processing. Satisfaction analysis to calculate prevalence and penetrance, gene frequency estimate using Hardy-Webber equilibrium. Chromosome mapping associated with lossy C7p 14.1, 13q21, q32, 19p 13.1.
Main resource analysis. Main findings. Prevalence 1.7 cases for 1 million live births. Family history. 13.4% of isolated cases had affect relative. Penetrance. 62.6% estimated for postsexual polydactyly. Gene frequencies.
Genetic analysis. Most cases follow an autosomal dominant inheritance pattern. Variability due to heterogeneity of loci. 7p 14.1 GLI3 13q p21 q32 PAPA2 19p 13.1, 13.2, PAPA3. So here we have some results and data collected of the type of polydactylia in in preaxial and potential with the type of a, a male or woman, and this another graph.
Parents, we have another statistics that shows the data results of polydactyl praxial without polydactyl, with polydactyl, the total. Then we have the characteristics, the cases, and the controls. And
Conclusion. Polydactyly is a cognition malformation characterized by presence of extra fingers or toes. Genetically, is a strong heritage component, most often following an autosomal dominant pattern with variable expression. The condition results from a mutation or alteration in several genes located in different chromosomes, notably 7p 14.1, GLI3, 13q21, Q32, PAPA2, and 19 P13.1, 13.2, PAPA3, showing that polydactyly is genetically heterogeneous disorder. Rich based on population data like a children's study, highlights the importance of genetic analysis, family history, and chromosomal mapping to better understand the heritage patterns and give genetic counseling for affected families. Overall, the study of polydactyly not only provides insight into human development genetics, but also contributes to the advance of clinical and genetic cognitive research. Finally, the bibliography.
and
And that
that's
is it.
it. Demasiado intelectuales.